Welcome to Biology My Passion. I am Soumya Harikrishna. Now we will see some previous year's questions from the chapter Molecular Basis of Inheritance. So the first question is, given below is one of the strands of DNA segment with the polarity 3' to 5' and the strand is TACG, 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 TACG. Write its complementary strand. So whenever we are writing the complementary strand, we need to write with the polarity. So what will be the polarity of the complementary strand? 5' to 3'. And here thymine is complementary to adenine. Adenine will be complementary to thymine. Cytosine means guanine. Guanine means cytosine. ATGC. So how many times it is repeating? 1, 2, 3, 4. So 4 times we can write ATGC, ATGC, ATGC 3'. Okay. Next, write a possible RNA strand that can be transcribed from the above DNA molecule formed. So this is the DNA molecule formed. So in this, by seeing the polarity, 3' to 5' is the template strand, 5' to 3' is a coding strand. Coding strand will have the similar sequences like the new RNA strand, and its polarity will be also the same, 5'. But only difference will be thymine will be uracil because newly formed one is RNA after transcription. So answer will be AUGC, AUGC, AUGC. One more time. 3'. See here we have to write only the complementary strand once, that is the coding strand. If only we have to change T into U wherever T comes. State a functional difference between the following codons, AUG and UAA. We know AUG is a start codon for translation process, and also it codes for methionine. It has dual function, that is another question. Whereas UAA, UAG or UGA, these three are stop codons. Okay, so start codon and a stop codon. They do not code for any amino acids, unlike the start codon, or these are called nonsense codons. Now coming to specific means what? One codon codes for only one amino acid. Degenerate means what? One amino acid can be coded by more than one codons. Write the start codon along with the amino acid it codes for. Just now we saw start codon is AUG. Amino acid it codes for is methionine. And any two stop codons, just now we discussed three, there are UAA, UGA and UAG. Given below is a segment of DNA strand. Write its complementary strand and the RNA strand that can be transcribed from the DNA molecule. So 5' to 3' is given, so it is 3'. Then what is it? The T will become A, T, G, G, C, A, C, T, G, C, A, G, 5'. Here is a small difference in the RNA strand. We cannot write a complementary to this because 5' to 3' strand is the coding strand. So the coding strand has the similar codons. Only thing is that I told thymine should be replaced by what? Uracil. So now what will we get? 5', U, A, C, C, G, U, G, A, C, G, U, C, 3'. The other two properties are given. Genetic code, degenerated, as I told, one codon, many amino acids. Universal means what? The codons code for the same amino acids irrespective of the organisms, whether it is prokaryote or eukaryote or higher organisms like human beings. So a schematic diagram of a transcription unit with the polarity of the DNA strands and label coding strand, template strand and a terminator. A transcription unit has got three parts. First part is called the promoter. Second part is a structural gene. Third part is a terminator. So there are two antiparallel strands in a DNA. The first strand is with the polarity 3' to 5'. So this strand is called a template strand. The second or the complementary strand with a 5' to 3', that is called a coding strand. Then 5' end or the uphill of the coding strand, there is the promoter. Whereas the downhill or the 3' end of the coding strand, there is the terminator for ending the process. Events occurring in a replication fork. Replication is fork is formed during DNA replication process. So first what happens is DNA dependent DNA polymerase enzyme catalyzes the polymerization reaction of deoxynucleotides or deoxyribonucleotide triphosphates. And the direction in which it is happening is 5' to 3'. On one strand, that is, suppose these are the two strands in the replication fork. One is 3' to 5', this is 3'. So here this is called a template strand with the polarity 3 to 5. So in this, the new strand will be forming continuously, that is, it is from 5 to 3. Whereas in this will be forming in a discontinuous manner, that is called a lagging strand. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase. So RNA polymerase, there is only one type required in the prokaryotes, whereas 3 types of RNA polymerases are required in eukaryotic cell. So they are 1, 2 and 3. You can remember in the order RMT. That is, 1 is for rRNA, 2 is for mRNA, 3 is for tRNA. So RNA polymerase 1 transcribes rRNA. They are 28S, 118S and 5.8S. Polymerase 2 transcribes precursor of mRNA, that is hnRNA, which is otherwise called a heterogeneous nuclear RNA. We know that initially when it is forming, it has exons, introns. Introns have to be removed by splicing. Then only we can get the mature RNA. Then RNA polymerase 3 transcribes tRNA, which includes 5 srRNA, snRNAs and small nuclear RNA. Explain the role of different genes in lactoperon when it is switched on state. Switched on state means what? Presence of lactose is there, which is an inducer. The regulator genes means here I is the regulator gene. This is the operator site. So regulator gene produces a repressor. So you can see that from the I gene, by transcription or repressor mRNA is forming. That repressor mRNA will become a protein by translation. That protein is called a repressor protein and inactivated in the presence of inducer, that is lactose. See here, since lactose is present in the midst in the on state, so the repressor protein is inactivated by the inducer. Inducer is nothing but the substrate, that is lactose here. RNA polymerase then gets access to the promoter site. So here there is no repressor protein binding here. So this is open. So the RNA polymerase enzyme can continue its work here because of the lack of repressor protein here. And the transcription can proceed. Here 3 structural genes are there, Z, Y and A. So you know that the Z is producing the enzyme that is called a beta galactosidase enzyme, which is needed for the hydrolysis of the substrate lactose into glucose and galactose. For that to come inside the cell, there needs permease enzyme, which will increase the permeability of the cell membrane to lactose, so the lactose can come inside the cell. That means the bacterial cell. A stands for the gene which is transcribing the transacetylase enzyme, and this transacetylase enzyme is needed for the process of metabolism of lactose. So usually the process is like this. From the regulator site I gene, there forms an mRNA which will produce a protein called a repressor protein. If this repressor protein is going and binding here, then the RNA polymerase will not be able to do the process of polymerization of this mRNA, that means by transcription or later translation. So none of these enzymes are forming in the absence of lactose. So this is called a off state. But when there is on, that means on means what? In the presence of an inducer. What is inducer? Inducer is lactose. So when lactose is there, this repressor protein is being made, but the lactose or the inducer will bind with it and changing the configuration. Now it is not able to bind to this operator site. So operator site is open. So the RNA polymerase can continue its transcription of Z, Y and A genes, producing these enzymes and later helping in the metabolism of lactose. This is called the on stage of lactopro. Explain the discovery made by Hershey and Chase using radioactive sulfur and phosphorus in their experiment. We know this was the experiment which unequivocally proved that DNA is the genetic material, because the first effort started by Griffith. Through his experiment, he confirmed transforming principle, and that experiment was continued by Oswald Avery, MacLeod and McCarthy, and they confirm that DNA is the material which transformed the non-virulent type into virulent type, but still the doubts were there. So who proved it, confirmed the DNA is the genetic material? The experiment by Hershey and Chase. Actually, they used viruses called bacteriophages. Virus attacking bacteria are called bacteriophages. They grew the virus into medium, that is, one is containing radioactive sulfur to make a protein called radioactive, because when a bacteriophage is considered, the outer coat and a genetic material, these two are the components of the virus. So outer coat is made up of protein which contains sulfur, whereas inner genetic material is DNA, which consists of what? Phosphorus. So if you grow them in the radioactive sulfur, this protein coat will take up the sulfur and becomes radioactive. And if they grow the virus in a medium containing radioactive phosphorus, which part will become radioactive? The DNA will become radioactive. Then later they saw which is going inside and making the bacteria radioactive. If it was the phosphorus found in the bacteria, so we found that DNA was making it radioactive. So they grew viruses or bacteriophages in a medium containing radioactive sulfur to make protein called radioactive, and grew bacteriophages in radioactive phosphorus medium to make DNA radioactive. Then they allowed the bacteriophage to infect Escherichia coli with these radioactive phages separately. And once the bacteria was being infected by the viruses, what did they do? They blended the mixture so that the protein coat would be removed. Then bacteria which were infected with the viruses with radioactive protein, or that means that contains radioactive sulfur, did not show any radioactivity on centrifugation. So after blending their centrifuge, they got two bands. One was the viral pellet and the viral coat. Second is bacterial pellet. So in the bacteria only radioactivity was seen. That showed and what was seen? Phosphorus. So that means the DNA part came inside the bacteria. The viral part was radioactive sulfur. So bacteria which were infected with viruses with radioactive DNA were radioactive, and they proved that DNA is the genetic material. Next is another important experiment in this chapter. Describe the experiment where Matthew Michelson and Franklin Stahl use heavy isotope of nitrogen. So what was this aim of this experiment? To prove semi-conservative method of replication. So aim you should know thoroughly. The name of the scientist is very important, and also the experimental material that they used, whether it was Escherichia coli or bacteriophage. Here in this case they used Escherichia coli, which replicate every 20 minutes. And the medium they used for growing was ammonium chloride, which contained heavy isotope of nitrogen. So initially they grew this bacteria Escherichia coli in ammonium chloride with a heavy isotope of nitrogen N15 medium for several generations. So N15 was incorporated into a newly synthesized heavy DNA. These cells were transferred into NH4Cl normal medium. So initially they made both the strands with the N15. That means heavy isotope of nitrogen by continuously growing the bacteria Escherichia coli in the ammonium chloride medium. Then DNA extracted from culture after one generation. So first they made it heavy. When they did centrifugation, what did they get? A heavy band at the bottom they got, because it was N50. Then they transferred it to ammonium chloride N14 containing medium, that is the normal nitrogen or light. So in this case they found that after 20 minutes, the bacteria replicate. That means the DNA also replicates. So DNA extracted from the culture after one generation, that is 20 minutes, it had a hybrid because these two separate out and a new strand is forming. The new strand from the medium N14. So one strand is N14, one strand is N15 in both the cases. So that is called a hybrid, that is with the intermediate density. So the band was seen or little above, that is N14 and N15. Then they continue to grow within the N14 containing medium ammonium chloride. So after 40 minutes was composed of equal amounts of hybrid and light DNA. Because you know, when these get separated, we get 4 strands like this. This is N15, this is also N15, and the rest are red are N14. Newly formed ones are N14. So this will have one N15, N14. This also will have one N14 and one N15. That is a hybrid. But these two are new light ones. They both are light isotope, or they are forming a band above this, that is N14 and N14. So the experiment proved that DNA replicates semi-conservatively, means one strand will be older, that is conserving half of the strand, the other one will be new. Instead of the above explanation, the following diagram also can be considered. So if you draw the proper diagram with all the labeling, see, labeling is very important. You have to write N15 DNA half mark. Here the generation 1 N15 and N14, that is half mark. Then here again this N14, N15 half mark. Then here also you are writing heavy isotope hybrid, it is half mark, and this hybrid also half mark. So all points you have marked. You have to write the timing also. So everything is marked properly, showing the band in the centrifuge. Then you will get the entire mark for this process. What are SNPs? Where are they located in human cell? State any 2 ways the discovery of SNPs can be of importance to humans. SNPs are single nucleotide polymorphism, locations where single base DNA differences occur in humans. For example, if there is a sequence ATGC, only one place it is changing, maybe ACCG or AGCG or ACG, like that, only one place if there is, that is called a single nucleotide polymorphism. Location is present in the human genome, and or we can write it is in the human chromosome. What is the importance? It is helping in finding chromosomal location for disease, associated sequences, and tracing human history. This we are making use of in DNA fingerprinting also. Write the features that biomolecule must fulfill to be able to act as genetic material. DNA and RNA are both genetic materials. Which one of the two is more stable and why? So first answer is the features of a biomolecule to be more able to act as a genetic material. So there is a discussion in the textbook which is the better genetic material, DNA or RNA. So these are the characteristics or the qualities that we consider. One is it should be able to replicate. In that sense, both DNA as well as RNA can replicate. Should be structurally and chemically stable. We know DNA is more stable and less reactive than RNA. Provides scope for slow change or mutations required for evolution. There should be scope for mutation or change, but it should be slow. Whereas in RNA, the mutation is very fast. Should be able to express itself in the form of Mendelian characters. That is also possible with the DNA. Now the question is, which is more stable, DNA or RNA, and why? So DNA is more stable. Why? Because DNA is first of all double-stranded, and there is only one OH group at the 3' end. There is no OH group at the 2' end. So second carbon atom we can say, and so it is chemically less reactive. More OH means OH is a functional group. More OH means more reactive. Here one OH is less means less reactive. Then presence of thymine instead of uracil also confers a stability to DNA. Next question is about DNA replication in prokaryotes, and write the phase in cell cycle where DNA replication occurs in a cell. What will happen if cell division fails to occur after DNA replication? Replication of DNA begins at a region of DNA called the origin of replication site. DNA dependent DNA polymerase enzyme helps in the polymerization of deoxynucleotides, which are the substrates. Replication occurs within a small opening called the replication fork. Polymerization takes place always in 5' to 3' direction only. And one strand grows in continuous manner, that is the template strand 3' to 5', while the other with the template strand 5' to 3', DNA strand grows in a discontinuous manner, forming small stretches called Okazaki fragments. Discontinuously synthesized fragments are later joined by the enzyme DNA ligase. So in which stage of cell division it is happening? During the S phase of the interphase of a cell cycle, DNA replication happens. And also, if the DNA after that, if they fail to segregate, what happens? It will result in polyploidy. Name the types of cells and the process by which hnRNA is formed. hnRNA is the precursor of mRNA, which is heterogeneous nuclear RNA, which is formed after transcription. Describe the processing mechanism it undergoes before it becomes functional. So hnRNA is formed in eukaryotic cells during the process of transcription. What is transcription? The information from a double-stranded DNA is copied into RNA, single-stranded RNA. Primary transcript, that means this hnRNA is the primary transcript which contains both exons. Exons are the coding sequences for proteins, and introns, which are non-functional or non-coding sequences. We can call them as intervening sequences. It is subjected to a process called a splicing. Because this exons and introns present, that gene is called a split gene. Split gene has to undergo a process called a splicing, where exons are joined by removing introns. Where introns are removed, exons are joined in a defined order. hnRNA, heterogeneous nuclear RNA, undergoes also additional processing like capping with a nucleotide like methyl guanosine triphosphate MGTP added to the 5' end of the hnRNA. And there is tailing also occurring at the 3' end by adding adenylate residue A like that. 200 to 300 adenylate residues will be added at the 3' end. So there are 3 post-transcriptional complications involved here. One is splicing, that is removal of introns. After that, capping at the 5' end with an unusual nucleotide, that is MGTP, and tailing at 3' end by adding 200 to 300 adenylate residues. How is 2.2 meters length of double helix mammalian DNA packed in a small nucleus of its cell? Explain. Positively charged histone proteins, due to presence of lysine and arginine, they are the two basic amino acids present in histone protein, form a histone octamer with the 8 molecules. Because of lysine and arginine, the protein is positive in nature, histone protein. It forms a histone octamer using 4 types of histones, H2A, H2B, HC and H4. These are the histones forming the histone octamer, and negatively charged DNA wraps around this histone octamer to form nucleosome. DNA being negatively charged can attach this positively charged histone proteins. Then a nucleosome contains 200 base pairs. So the length required for this winding is 200 base pairs of DNA helix. Nucleosome constitute repeating units of structure called a chromatin, appears as beads on a string-like structure. So at the end it looks like this, a chain of DNA, and octamer looks like the beads. Then further coiling is done by the non-histone chromosomal proteins. Differentiate between euchromatin and heterochromatin, giving one structural and functional difference only. Euchromatin means a loosely packed, structurally it is loosely packed, whereas heterochromatin is a densely packed. So as a result of this, there can be one more difference. Loosely packed will stain less, whereas heterochromatin being tightly packed will stain darker. Then function, if you see, transcriptionally active, that means we can transcribe it and make proteins, whereas transcriptionally inactive, making protein is not possible. Next question is about the process of transcription in prokaryotes. So first, DNA dependent RNA polymerase enzyme is used for transcription, because in replication it was DNA dependent DNA polymerase. This catalyzes the process of transcription. There are 3 steps involved: initiation, elongation and termination. What is initiation? That is beginning of the process. RNA polymerase is the enzyme which will bind to the promoter region. Where is promoter region located? 5' of the coding strand, associated with the initiation factor sigma factor at the 5' end of the DNA. The second process is elongation. So once the process begins with the sigma factor associated with the RNA polymerase, now the nucleoside triphosphates, because they are the substrates which will be acting as substrates, will be added to the DNA strand based on the information on the DNA strand following rule of complementarity. What is rule of complementarity? Adenine base pairs with a uracil and guanine base pairs with a cytosine, because here it is uracil, the newly formed structure or the biomolecule is RNA. RNA polymerase help in opening the helix and continuous elongation, and finally when it reaches the other end, that is 3' end of the coding strand, termination happens by the attachment of another termination factor called a Rho factor. As a result, the nascent RNA falls off. Nascent RNA means the newly formed RNA falls off along with the enzyme. Enzyme also gets separated, completing the process of transcription. Here you can see the diagram. This is a textbook diagram showing 3 processes: initiation, elongation and termination. Next is the first experiment in the biological world to prove that the genetic material nature. Write the scientific name of the organism Griffith used in his experiment. So Griffith used a Streptococcus pneumoniae bacteria. It is the bacterium which can cause a disease called pneumonia. State the conclusion he arrived at. So he concluded that R strain bacteria has been transformed by heat-killed S strain bacteria. Actually, if you remember the experiment, he used 2 types of Streptococcus pneumoniae, R strain and S strain. R strain is the one which is the rough strain, and the rough strain is a non-virulent, or it cannot cause pneumonia. Whereas S strain is having a smooth polysaccharide coat. As a result, it is the virulent type which can cause the disease. He injected them into the mice. First, when he injected R strain, the mice lived because there is no disease. When S strain was injected, the mice died due to pneumonia. Then what he did, he heat killed the S strain and injected. Then also mice survived because no pneumonia was caused by the heat killed S. But when he mixed this heat killed S, which is non-virulent, with the R, which is also non-virulent but it is live, but he found that mice died. When he did the autopsy, he found that the mice died of pneumonia, and he could find live S virulent strains in the body of the dead mice. So from where this live S came, because he didn't inject the live S once, so he found that the S, which is heat kill, somehow transformed the non-virulent R into virulent S. Okay, so he concluded that R strain bacteria has been transformed by the heat killed S strain bacteria, or R strain became virulent from non-virulent. He proved the transforming principle of genetic material here. Name the scientists and describe how they experimentally proved the biochemical nature of the genetic material, which was earlier considered as protein. So Griffith stopped his experiments with his conclusion of transforming principle. Still he could not tell which is the genetic material precisely. Then this experiment was continued by 3 other scientists, Oswald, Avery, MacLeod and McCarthy. They purified biochemicals like proteins, DNA, RNA from heat-killed S cells to see which one could transform live R cells into S cells. One by one they isolated and introduced into the body of the bacterium. They also discovered that protein digesting enzymes like proteases or RNA digesting enzymes or RNAs were added. It did not affect the process of transformation, because even if protein is digested or RNA is digested, still the transformation was happening. But when DNA's enzyme was added, DNA got digested. After that, he could not find any transformation. So they concluded that DNA is the genetic material. Here what happened? Digestion with the DNA-ase enzyme did inhibit transformation, suggesting that DNA caused the transformation. So this question, first part we already discussed. How did Michelson and Stahl reach the conclusion of semi-conservative method of replication? I am not repeating that, but explain the contribution of Taylor et al., means Taylor and his colleagues, in DNA replication in flowering plants. So the confirmation or another experiment in this field was done by Taylor. So they worked on plant called a Vicia faba or faba beans. What did they do? They used a radioactive thymidine to prove that DNA in the chromosomes also replicates semi-conservatively. Write the steps that lead to amino acylation of tRNA in the cell. See, prior to translation, there is a process called amino acylation of tRNA or charging of amino acid. These processes are the initial processes happening during translation. So during that time, what happens is, and amino acids are activated by adding ATP to it, because it is an energy requiring process, that is adding amino acids or connecting them with a peptide bond. So the energy needed is derived from ATP. So initially amino acid will combine with ATP to form amino acid and a ATP complex, and that is called a charged amino acid. So then it is linked to the corresponding tRNA, that's called a cognate tRNA, because each tRNA is very specific about the amino acid it can attach with. So once the tRNA attaches with already charged amino acid, that is called the amino acylation of tRNA or charging of tRNA. Explain the events that occur in ribosomes during translation in prokaryotes. So the third process in central dogma is a translation, where from a mature mRNA, processed mRNA, how the protein is formed. So the ribosome is the protein factory, right? So ribosomes bind to the mRNA at the start codon or AUG, that is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase. Once the initiation starts by binding of the tRNA or the initiator tRNA with the AUG codon of the mRNA, the two subunits of the ribosome also come and attach, and the process starts. So during elongation phase of protein synthesis, this stage complexes composed of amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA. Means based on the sequences or the codons on the mRNA, more tRNA will come, that cognitive tRNA with the corresponding amino acid, and will be attaching the amino acid with the previous one using a peptide bond, forming complementary base pairs with the tRNA anticodon. Ribosome moves from codon to codon along with mRNA, and amino acids are added one by one, then translated into polypeptide sequence represented by mRNA. At the end, a release factor is binding to release the process or the stop the process, because stop codon is there, UAA, UGA or UAG, terminating translation and releasing complete polypeptide from the ribosome. So initiation is by the mRNA is encountering two subunits, smaller subunit of ribosome. A larger one also comes and binds with it, and then the initiator tRNA comes and binds. During elongation, different tRNA with the corresponding amino acid will be coming and binding according to the complementarity rule or according to the information on the mRNA, and they are connected by peptide bond. At the end, when termination happens, the release factor binds to the terminator codon, and once it reaches the terminator codon, and as a result the process stops. For the detailed video, you can visit Biology My Passion. I'll give the link in the description box. Name the types of DNA that forms the basis of DNA fingerprinting and mention two features of this DNA. So usually satellite DNA or repetitive DNA are the basis of DNA fingerprinting process. These sequences normally don't code for any proteins. They are showing high degree of polymorphism. Write the steps carried out in the process of DNA fingerprinting technique and mention its applications. Processes are somewhat similar to rDNA technology in the beginning. That is, first isolation of DNA, then digestion of DNA using restriction enzymes, that is restriction endonuclease, separation of DNA fragments by gel electrophoresis. Up to that is same like rDNA technology. Then after this, we transfer it to a blotting paper using southern blotting technique, and we get it in a nitrocellulose or nylon membrane. After that, we use radio labeled VNTR, variable number tandem repeats, probes for hybridizing with this DNA, and detection of hybridized DNA fragments are done using orthoradiography. So the applications, if asked, DNA fingerprinting is widely used in forensic science. It's also used in determining population and genetic diversity. It is used in paternity test, and also it is used in study of fossils or in evolutionary studies. Here again translation question, but it is asked in a different way. Explain the relationship of ribosomes, which are the protein factory, tRNA, which are actually the adapter molecules, and a mRNA, which has the information during the process of translation in prokaryotes. So amino acids are activated in the presence of ATP. I told first charging of amino acid are linked to the cognitive DNA or charging the DNA, amino acylation of tRNA, that's the first process. Then ribosome is a cellular factory for protein synthesis, which exists as two subunits. For initiation or the starting of the process, small subunit of the ribosome binds to mRNA. So first smaller subunit comes and binds with it, then larger subunit also comes and binds with it. then based on the codon, the tRNA with the anticodon will be coming. The first one will be with the methionine. So the larger subunit has two sites for the subsequent amino acid to bind to each other with a peptide bond. That is, ribosome also serves as a catalyst for the formation of peptide bond. Ribosome proceeds to elongation phase where charged tRNA sequentially bind to the appropriate codon in mRNA by forming a complementary base pair. That means the amino acids will be connected by peptide bonds and the tRNA anticodon. Ribosome moves codon by codon along the mRNA. Amino acids are added one by one. At the end, a release factor will come and bind with the stop codon, either of any of this UAA, UAG or UGA, and the process will terminate. You have to entirely understand the process very well. Describe the structure of a transcription unit. I am not repeating, we have already done that. Explain the basis of defining two DNA strands of a structural gene in a transcription unit. You know that the template strand is the one with the polarity 3' to 5', which will be transcribed, whereas the other strand with the 5' to 3' polarity is called a coding strand, which has all the codes similar to that of the mRNA transcribed. And all the references are made with respect to the coding strand. Explain two complexities that are observed in the process of transcription in eukaryotes and not in prokaryotes. So there are three RNA polymerase in the nucleus. RNA polymerase 1 transcribes rRNA, 2 mRNA and 3 tRNA. I have told you to remember as RMT. That's the first complication, because in case of prokaryotes there is only one RNA polymerase, whereas here three RNA polymerases transcribing three different RNAs. Now the next complication is processing of the initial transcript. That is, the primary transcript is called a hnRNA. It contains both the exons and non-functional introns subjected to splicing, where introns are removed, exons are joined in a defined order. hnRNA undergoes processing called capping and tailing. In capping, nucleotide that is mGTP is added to 5' end, and tailing, adenylate residues are added at the 3' end in template independent manner. This fully processed hnRNA is now called a mRNA and will be transported out of the nucleus for translation. So this can be asked as a different way. What are the post-transcriptional modification? So there you have to say it has to undergo splicing, then capping and tailing. Complexity, if asked, compared to prokaryotes, that is RNA polymerase also you have to mention. List any four major goals of human genome projects. The goals are listed in the NCERT as such. 1. Identify all the approximately 20,000 to 25,000 genes in human DNA. Determine the sequence of the 3 billion chemical base pairs that make up human DNA. Store this information in databases. Improve tools for data analysis. Transfer related technologies to other sectors such as industries. Address the ethical, legal and social issues (ELSI) that may arise from the project. So total 6 goals are there. Any 4 ways the knowledge from HGP is significant for humans. How can HGP help us? Solving challenges in healthcare, agriculture, energy production, environmental remediation, diagnose disorder, treat disorders, prevent disorders, any 4 of this. And also the 2 vectors used in HGP were BAC and YAC. BAC only asked here, that is bacterial artificial chromosome, and YAC also was used as a vector, yeast artificial chromosome. So they were used as vector for cloning of DNA fragments. We already discussed the on position of lac operon. List the different components of lac operon is asked here. Explain the role of these components when this operon is in open state. So here the components are, one is the regulatory gene, which is otherwise called the I gene or the inhibitor, and 3 structural genes Z, Y and A. Repressor gene, which is synthesized all the time constitutively from the I gene, is inactivated by interaction with the inducer, lactose or allolactose. So in the presence of the inducer, that is lactose mostly or allolactose, it will bind with the repressor protein and does not allow the repressor protein to bind with the regulator site. This allows RNA polymerase access to the promoter. Since there is no repressor, the RNA polymerase can move through it and turning on the transcription of these genes. 3 genes are Z, Y and A, which in turn producing enzymes responsible for digestion of lactose. So which are the enzymes? Beta-galactosidase for the metabolism of or breakdown of lactose to glucose and galactose. Then Y gene will code for permease and A gene for transacetylase. Entire diagram also considered in layer of this explanation, but you have to mention each and every component and the functions. Hope you understood all these concepts we discussed today. Apart from this, there are a lot of 2 marks and 3 mark questions are coming in that chapter. So please go through the chapter very well and prepare well for your exam. All the best. Thank you for watching Biology My Passion.